and let me go to the next speaker, who is Professor Daniel Belsky from Columbia University, and he will be talking about testing health disparities in cognitive and biological aging in older adults in the United States. So Dan, go ahead.
Well, thank you very much for having me. And just to provide some orientation to your audience, I'm not, in fact, in a closet. This is my children's art room, which has become my office for this pandemic. And it doesn't hide well behind a Zoom background because of the heterogeneity and textures and colors and so forth. So you can enjoy some artwork and some art supplies as a background to my presentation. I'm going to go ahead and share my screen and get started. I should say, as a disclaimer, this talk is mainly about disparities in biological aging and what they may mean, rather than cognitive aging, although that's a direction we are moving in. So I'm going to try and live up to the title to the best of my ability, but mostly what I want to do today is talk with you about how we've thought about the problem of trying to understand disparities in aging, and I think it segues very nicely from the end of the comments section in Heather's presentation, where the question of what is driving these disparities is coming up. And that's sort of where we begin and how we think about the challenges associated with asking this question. So before I get started, I want to acknowledge our funders, but also my collaborators for their intellectual contributions to the work you're about to see, as well as their substantive contributions in terms of actually getting the work done. Colleagues and co-investigators, Jen Manley, Peter Munich, Jackie Taylor at Columbia, Kathy Harris, Allison Aiello, and Claire Yang at UNC, Kathy Whittam at John Jay, wonderful postdoctoral scholars in the Manley Lab, Indira Turney and Justina Avia Reiger, Laurel Raffington from the University of Texas, and then my team at Columbia, Gloria Graf, Miraj Kathari, and Dayan Kwan, who's since gone to UCLA. So I'll begin just sort of orienting to you to sort of how I think about aging. For the purpose of this presentation, we're going to talk about aging as a biological process. It's obviously a process of human development involving sort of personologic growth and change. But I'm here focused on this progressive decline in system integrity that occurs as we grow older, which ultimately disables and kills us. And in recent years, we've come to understand that this progressive decline in system integrity arises from an age-dependent accumulation of molecular changes that ultimately impair resilience and drive damage to our various organ systems, making us sick. This cartoon model, which I'll use a bit, describes the pathway envisioned in the geroscience hypothesis, which has guided much of my work over the past several years, in which this accumulation of molecular changes that occurs with aging drives decline in system integrity, which in turn leads to functional decline, disease, disability, and death. And of course, the geroscience hypothesis is that intervention on these molecular changes may be able to forestall or prevent these declines in system integrity, extending healthy lifespan. We have, I guess, a default model often when we start talking about aging to think about it as a process unfolding at the very end of the lifespan. But of course, we all know that what happens to us at the end of life is in important ways conditioned by the exposures we accumulate from the very beginnings of our development. And ultimately, we think about biological aging as a process connecting our exposure history with the development of disease, disability, and ultimately the causes of death. And so just as geroscience positions the molecular changes that accumulate with aging as an opportunity for intervention, we're starting to understand them as a point of intersection between our exposure histories and the development of aging-related disease. And that has suggested to me and to many of my colleagues this health equity geroscience model in which we can understand social determinants of health as sitting upstream in many ways of some of these molecular changes, not as their only causes, but as modifiers of the rate at which they accumulate. And this figure comes from a recent review by Eileen Crimmins, and there are several of these social determinants of health listed, and I should just expand on this idea that, while low socioeconomic status, or SES, in the figure, and minority status are represented as boxes, they themselves reflect a range of different exposures, including, as Heather suggested, psychological stress, but also differential burden of exposure to environmental toxicants, differential access to health-enhancing resources, and so on. So we've done some work on linking these social determinants of health to measurements of biological processes of aging, mainly focused on early life and adult measures of socioeconomic status, but also some analysis of how adverse life events, particularly victimization and maltreatment in childhood, may structure a faster pace of aging through adult life. And in fact, in Laurel Raffington's paper, which will come out in a month or so in Pediatrics, we find evidence that this process of acceleration of aging may occur even prior to the onset of puberty. So we see signs of accelerated biological aging in children exposed to socioeconomic adversity. And I think we'll be seeing a lot more work on this topic. NIA has funded, in collaboration with NIMHD and others, a range of projects to investigate genomic mediators of health disparities. And this is going to produce a lot of data that can link, in particular, DNA methylation, but also gene expression and other molecular changes with the exposures that we think drive health disparities, and also with the outcomes as measured. I note that in this little diagram I'm showing you, we haven't done work specifically on Eileen's minority status box. So we haven't done a lot to compare biological aging, in particular between Black and white Americans. And that's a pretty significant omission, given that the disparities we see in the health of Black and white Americans are profound and drive a substantial degree of the social gradient in health that we observe in our country. It's not the case that nobody has bothered to look. These are three papers of varying degrees of recency that have attempted to address this question. But one of the reasons that we have hung back and tried to do some more methodological development is that there are a number of challenges that face us when we attempt to map molecular measurements, in particular, onto differences between Black and white Americans. And I want to talk about sort of two issues that arise from the fact that the development and validation of these measures of biological aging has taken place in nearly exclusively white samples. So it's not the case that there are no non-white data contributing to the development and validation of measures of biological aging, but they represent a distinct minority of the data that are contributing to these models. And there are two consequences of that disparity in representation of different populations in the development of these measurements. The first is sort of a pure measurement artifact consideration. The social construct of race and all the exposures that come with it overlaps to an extent with differences in genetic ancestry. And there are genetic artifacts in many of the kinds of genomic data in particular, but also potentially in more conventional biomarker data that we measure. And so the inference we make about how a DNA methylation state, or potentially even a blood chemistry analyte, relates to disease pathogenesis may be disrupted by the presence of genetic variants that are not related to differences in health, but may affect the measurement of that intermediate quantity, the measurement of the DNA methylation state, or the measurement of the blood chemistry analyte. And that can contribute noise in a system, and it may also contribute bias. The second consideration is that the kinds of exposures that we understand to drive processes of aging differ between groups, let's say Black and white Americans, not only in their volume, but also in their kind. And in thinking about this, I've been considering recently Kate McLaughlin's model of threat versus deprivation as a source of stress in people's lives and impacts on their biology. And so the kinds of stresses that make up the health effects of systemic racism are different in kind than the kinds of stresses that drive the health effects of poverty, absent that component of systemic and structural racism. And so it may be the case that if we develop our measurements in populations that have little or no exposure to certain causes, those measurements may be less sensitive to the impacts of those exposures on an aging process. And that can similarly bias the kinds of measurements we might seek to make of disparities. A second issue that arises is that we know, for example, that Black Americans live shorter lives than white Americans. And as a consequence, if we're studying the process of aging, it means the farther we go in the lifespan, the more pronounced essentially selection bias in our studies will become. And this is something we encountered in some work with Daniel Parker, who's a physician and scientist here at Duke, working with the Duke EPIs cohort, which Heather introduced you to in her talk, where we found that a panel of blood chemistry-based measures of biological aging were substantially less predictive of mortality in the Black as compared to white participants in the EPIs. And part of the challenge, we think, is that these individuals gave their blood when they were all north of age 70. At that point, the white participants, and these are North Carolina-born study participants, had outlived the life expectancy for their birth cohort by a few years. The Black participants had outlived life expectancy for their birth cohort by more than a decade. So they were in some sense already exceptional agers by the time we began to observe them. And that's going to make challenging inferences about differences in both the level of measurements of biological aging, but also the criterion validity of those measures. Okay, so what I'm going to share with you first today, and I'll spend most of my time on, is some work that we undertook to sort of take on this project and ask in a rigorous way whether it was possible to locate available measures of biological aging in between the sort of social determinants of health exposures we're interested in this particular case, Black and white racial identity, and disparities in healthy aging. And this is work led by Gloria Graff, who is now my PhD student. So the data we're drawing from here come from the US Health and Retirement Studies 2016 venous blood study. This is one of the first data sets to produce a large scale DNA methylation and blood chemistry database for a diverse sample of Americans. And in our case, we're interested in older adults for this initial study. So that's where we focus our attention. I think I want to orient you, though, to some differences that confront us, from the beginning. Among them, that the rate of participation in the venous blood study, and in particular in completion of provision of data, is different for Black and white participants. So you can see that about 65% of white versus 55% of Black participants are contributing data to the study. So there may already be some selection bias built in. And we expect that this is going to be non-trivial with respect to our exposure of interest, which are the stresses of racism, because a key driver of non-participation of Black research participants in blood and molecular data collection is the sort of racist history of American medicine and the exploitation of Black research participants. So that sits in the background of all this, and we don't yet have good models to try and correct for that, but it is something that we're thinking about and trying to work on. These are the data that we're looking at, and I just want to orient you to some numbers, which I generally don't like to put all over my slides, because the sample sizes for the different measurements we're able to analyze are different. So the venous blood study collected blood and did blood chemistry analysis on about 10,000 people, but only about 4,000 of those individuals were included in DNA methylation analysis. And so I'm showing you comparisons of age, sex, and a sort of minimal educational level in each of those groups, so you can get a sense of how the participants differ. In general, the methylation sample is representative of the venous blood study sample, but that sample is younger, better educated, and whiter than the US Health and Retirement Study overall. To talk briefly about measurements with you now, we're going to deploy 3 blood chemistry measurements of biological aging, which we've studied in a number of papers, and the citations at the bottom of the screen are actually for the measures themselves, not for our work on them. I've used them together in a number of projects because they measure aging in different ways and make different assumptions about how that measurement process should unfold. And so when I see parallel results across these three measures, I feel confident that what we're observing is in fact a substantive difference in the physiology of aging. So the Clamara-Duval method is a method which essentially represents aging as differences in physiology between older and younger people. So you are the biological age at which your physiology would appear normal in a cross-section of a reference sample. And we use, for all of these, the National Health and Nutrition Examination surveys. Homeostatic dysregulation is a measure proposed by my colleague Alan Cohen, and it's a biomarker Mahalanobis distance. And as we use it, we essentially are quantifying deviation of a biomarker panel from a young, healthy reference, again, derived from the NHANES. It doesn't make any assumptions about linearity, and it does take into account the way in which biomarkers are correlated with one another in younger people and the extent to which those correlations break down as people age. And then the final measure is a blood chemistry measure of phenotypic age that was developed by Morgan Levine on her way to developing a DNA methylation clock of the same name, in which the criterion is mortality, and the measurement models mortality from a panel of biomarkers, in this case derived from machine learning analysis of 42 different blood chemistry analytes and complete blood count parameters in the NHANES. And so you can interpret this pheno-age metric as being the chronological age at which your biomarker predicted mortality would be approximately normal in the NHANES. We implement these in the HRS data using an R package that we put together that essentially allows us to draw new models from NHANES data using whatever biomarkers are available in our target sample. In this case, we used the biomarkers from Morgan's pheno-age because all of those were available within the HRS. And the package actually runs through a bunch of analyses that not only parameterize models that you can apply in your own data, and it will project them onto your own data as well, but it also validates them within the NHANES, comparing their prediction of mortality, disease, and disability to originally published versions of these algorithms developed within the NHANES, and also evaluates parallel criterion validity between older and younger participants, between men and women, between Black and white participants. So for those of you who are interested in playing around with biomarker data, it's on GitHub and available for you to use. In parallel to these blood chemistry measures, we're also analyzing a set of 5 DNA methylation measures. And I'll divide these into 3 categories. I think people by now are very familiar with the epigenetic clocks. These are DNA methylation predictions of age, and the interpretation that's given is that if your DNA methylation predicted age is older than your chronological age, then you're aging faster than we'd like you to be, and you're closer to death. I'll divide these into these 2 generations. The first generation clocks were developed by comparisons of older and younger people. And so like the Clamara-Duval biological age, the predicted ages reflect the age at which your DNA methylation state would be approximately normal in the reference samples that were used. And in fact, the reference samples for these clocks are a little peculiar, and they're described in our paper if you're curious. In part, following recognition of some of the limitations of these first generation clocks, in particular for predicting disease, disability, and mortality, and also their comparative lack of sensitivity to exposures that we know drive accelerated aging measured from physiology or functional decline, a second generation of clocks were developed based not on age alone, but on prediction of mortality. And both the Grimm age clock and pheno age clock that we analyze in this paper additionally incorporate DNA methylation modeling of physiologic states into that mortality prediction. And then the third category is a measure that we developed recently based on some work we've done studying changes in physiologies of research participants in the Dunedin study. And this is work with the Moffitt-Caspi lab, who are, for those of you who are in SSRI at the moment, just across the street. The pace of aging is the rate of within-person change in system integrity occurring over a period of time. In our study, it was 12 years of follow-up in a cohort that was entirely age 26 at baseline, and who were then followed up at 32 and 38 years of age. This multi-system rate of decline was then distilled into a DNA methylation measure, which provides from a single cross-section of data an estimate of how fast a body has been changing over the past decade or so. And in a bit of shameless self-promotion, I'll just put up this slide advertising our paper and also open source code you can use to implement our measure in your data, and also to acknowledge my collaborators, Terry Moffat, Afshan Caspi, David Corcoran, Karen Sugden, and David's programmer, Joey Prinz, who helped design and execute this study. This is a matrix illustrating how these different measures of aging are related to one another. And I don't imagine you have time to pore over it at the moment, but just to orient you, the upper right of the matrix can be read as a standard correlation matrix. The intersection of two measures gives their Pearson correlation. And then the lower left of the matrix essentially shows the scatter plot, again, of the intersecting measurements. And the only thing I'll note on this slide is that our blood chemistry measures of biological aging show a high degree of correlation in our health and retirement study participants. Our DNA methylation measures of biological aging show moderate correlation. And interestingly, and this is a sort of a productive development in the field, whereas you see correlations between the first generation epigenetic clocks and the blood chemistry measures of biological aging are what I would call low, correlations between this second generation of epigenetic clocks as well as our pace of aging measure and these blood chemistry measures are somewhat higher, suggesting that there may be some evidence that we're tapping a similar construct of aging. These are correlation matrices produced separately for black and white HRS participants. Again, as I mentioned, almost all of these measurements were developed in overwhelmingly white samples or exclusively white samples. And you can see that the correlations between the measures are relatively consistent. That's especially true of the blood chemistry measures. It is somewhat less true of the DNA methylation measures, which are the measures that have depended on almost entirely European ancestry populations for their derivation. And that may give us a hint at some limitations of their application in other populations. These are the dependent variables we're going to use to quantify healthy aging in the health and retirement study and give our measurements a test of criterion validity. They should be familiar to all of you. There are some functional tests that were given. There's peak flow, grip strength, tandem balance, and a gait test. There are activities of daily living, and there are diagnosed chronic diseases. This is a sort of a quick graph of the disparities. We're actually going to analyze counts of these problems, but here I'm just showing you proportion of black and white participants reporting one or more of each of these health deficits. And this is adjusted for the sampling design of the HRS. So it's essentially adjusting for age when you're seeing this, because the black HRS sample is a bit younger than the white HRS sample. So substantial differences as we're familiar with. And here's our basic test of criterion validity of these measures. The y-axis is showing you effect sizes in terms of incident rate ratios estimated from Poisson regressions for a 1 standard deviation unit change in the biological aging measure of interest. And so just to orient you to what we're seeing here, pretty consistent effect sizes for the blood chemistry measures, more heterogeneous effect sizes for the DNA methylation measures, with the grim age clock and our pace of aging measure showing somewhat larger effect sizes than the other measures, although there's some variability across these 3 sets of outcomes. Here's our test of parallel criterion validity, which is something that our work with Daniel Parker alerted us was very important. And if you saw me present in this meeting a couple of years ago at GSA, I was very concerned about some differences we were seeing in effect sizes within the HRS population. In the work that we've done in Gloria's project, I've become somewhat less concerned. and I'll sort of explain why. We do see modestly larger effect sizes for prediction of each of these healthy aging endpoints in white as compared to Black participants. However, most of those differences essentially reflect a higher overall baseline level of each of these health problems in Black as compared to white samples. And I'm sorry, I shouldn't say baseline, I should say population-wide. So what you can see in these graphs is that while the red slope is a bit shallower than the blue slope, it's because its intercept is a bit higher. And so at the most advanced or fastest biological aging, the predictions are quite similar. And this nevertheless did concern us until we re-examined the differences in effect sizes for chronological age, which is a measurement we think should be unbiased by the kinds of artifacts that I discussed earlier. We see the same differences in effect size for, I think in this case, it's a 5-year increment in chronological age that we're seeing for these differences in age-independent biological aging. And so I think what that suggests is that the differences in effect size are likely due to non-aging causes of disease that are more prevalent in Black as compared to white Americans. And our mind went to accidents and injuries, which can affect risk for certain diseases as well as mobility and function, but which may not directly intersect the biology of aging. Here now is the comparison of distributions of biological aging measures between Black and white participants. And what you can see, on the top row is very clear and consistent disparities in blood chemistry measures of biological age between Black and white participants. The Black participants, conditional on being the same chronological age, are biologically looking older. That difference is less consistently represented in the DNA methylation data, although the Grimm age clock and our pace of aging measure show the expected and large difference. And if you can squint at your screen, you can see we've provided the Cohen's d, so standard deviation difference between groups measure, and these are in the realm of sort of medium effect sizes, a Cohen's d of 0.3 to 0.4, or even as high as 0.5 for some of the blood chemistry measures. So difference is consistent with what we would expect given the magnitude of differences in the health outcomes we're observing. And so what we then do in the paper is we sort of fit these crude mediation models. The mediation models themselves aren't crude, but because these are cross-sectional data, we can't make any strong causal interpretation. It could be the case that it's in fact the health differences that we're measuring that are driving the biological aging. But we see that blood chemistry measures, the Grimm age clock, and our pace of aging measure are accounting for between 1/5 and 1/2 of the Black-white disparities we see in functional impairments, in activities of daily living limitations, and in chronic conditions. Okay, I'm going to spend very little time on the cognition data because it's really sort of a preliminary step. We're modeling change in cognition in the HRS, which have accumulated quite a lot of longitudinal data on this sort of 35-point composite scale composed of these two items or two sets of items. And it of course, shows very large disparities between the Black and white participants. I should note that, we're going to focus on disparities in rates of change rather than in level, because I think there are enduring concerns about how should we interpret these differences in level of cognition between Black and white adults. And that's something that we'll have more on in time to come. These are just some simple plots of the association between those rates of change and our biological aging measures. I'm just using the pheno-age blood chemistry measure here as a representation of the panel, since those are very similar in these data. And what we see are substantially weaker correlations than what we observe with the below-the-neck measurements of health. And so I think that raises some questions for the extent to which our model of the production of health disparities will translate from our analysis of below-the-neck health and mortality to the aging brain. So to summarize, I think we have several blood chemistry and biological aging measures that have basic proof of concept as tools for investigating health disparities in aging. I want to emphasize that that said, these concerns about measurement artifacts are not ignorable. And as I noted, the translation of the model to cognition may not be entirely straightforward. I want to leave you with this what I see as sort of the next steps in this health equity geroscience business, which are to refine causal inference with analysis of longitudinal change within individual lives, with natural experiment studies of exogenous change in people's circumstances, and with actual experiments. And we should have more on all three of those fronts for you in the next year. And then finally, I want to say that there is a developmental question to be addressed here in linking social determinants to biological aging with respect to when in the life course and at what level of biological organization these social determinants are impacting aging biology.
Okay, thank you, Daniel, for a very interesting talk. And we are running out of time, so I have to postpone all questions until after the last presentation.